We are now going to look at membrane potentials. This lecture requires a lot of your attention, so try to follow. If I lose you, just stop me and I'll repeat. What you're going to learn here, we are going to define what are membrane potentials. We are also going to look at the varieties of membrane potentials. We are going to state the ionic basis for what we call resting membrane potential. Then we are also going to explain how action potentials are generated. And lastly, we are going to outline the properties of action potentials. Action potentials are the ones we are calling propagated potentials. So this is our agenda this morning. Let's begin by defining what membrane potentials are. Membrane potentials just refers to electrical charges that exist across the cell membrane. Electrical charges that exist across the cell membrane is what we are calling membrane potentials. So that means that there are some charges that exist across the cell membrane. The cell membrane is not neutral in terms of electrical charges. These charges are usually measured in millivolts. So the unit of measurement of the electrical charges across the cell membrane is millivoltage. How do these charges come about? These charges come about because of the asymmetric distribution of charged particles across the cell membrane. We have just seen in the previous session that potassium is a positively charged ion and it is more inside the cell compared to outside. We have also seen that sodium is a positively charged ion, but it is more outside the cell than inside the cell. We've noted also that chloride is a negatively charged ion, and it is more outside the cell than inside. Likewise, we've mentioned something about the proteins, which are negatively charged ions, and they are more inside than outside. So if you look at those ions I've talked about, they are charged either positively or negatively, but they are not symmetrically distributed across the cell membrane. There are those which are more on one side than another. So it's because of this asymmetric distribution of charged particles across the cell membrane, it will then lead to the membrane having some charges across it. Membrane potentials are created by this asymmetric distribution of charged particles. There are three types of cell membrane potentials. We have what we call the resting membrane potential. We have what we call the graded potentials. And we have what we call action potentials. Action potentials are also known as propagated potentials. So I want us to then look at these three types of membrane potentials in the order in which I've listed them. That means we can begin with resting membrane potential. If I was to define for you the resting membrane potential, I will say it is the overall charge that exists across the cell membrane when the cell membrane is not disturbed, when the cell membrane is at rest. It is the overall charge that exists across the cell membrane when the cell membrane is not disturbed. The type of disturbance I'm talking about here is electrical disturbance. So there's nothing that is disturbing its equilibrium. It is the equilibrium that we achieve, that cell membranes achieve. As I've mentioned, it will be measured in millivolts. So that overall charge is measured in millivolts. This is the cell membrane. So we see charges here. Maybe you're wondering, why am I putting positive here and negative here? We are going to see that shortly. But what I want you to pick at this point is that there's some charges either way across the membrane. And that can be measured in millivolts. So as long as this cell membrane is not disturbed, the charges that exist across it will be termed the resting membrane potential. I want us to now look at how the resting membrane potential is generated. How is the resting membrane potential generated? One concept that I want you to build from now henceforth is that the cell membrane is permeable to potassium, to sodium, and to chloride. This permeability is through their leaky channels. We said leaky channels are channels which are always open. If leaky channels are always open, then it means that ions can freely move through these leaky channels from regions of higher concentration to regions of lower concentration. Having said so, remember that potassium is more inside the cell, sodium is more outside the cell, chloride is more outside the cell, but they are free to move through their leaky channels to the other compartments where the concentration is less. The images here show you those leaky channels. The leaky channels are usually specific, as we agreed last time. So you'll have potassium leaky channels, sodium leaky channels, and chloride leaky channels. Now let's see how these molecules then will be leaking. Let's consider this to be the cell membrane, and this side to be the extracellular fluid, this side to be the intracellular fluid. Let's also consider that this is the potassium leaky channel, this is sodium leaky channel, and this is chloride leaky channel. I want to see how potassium will leak through here. And especially, I want to ask myself the direction of that leakage. And this I want one of you to tell me. So if potassium was to leak, in which direction will potassium leak? Anyone?
Towards the extracellular fluid.
Thank you, Linda. Potassium will move outwards. The reason why potassium moves outwards is because potassium is more inside the cell than outside the cell. Right. How about sodium? How will sodium move? Anyone?
Sodium will move inside to the
towards intercellular fluid.
Very good. So sodium will move inwards, and that will also be similar to chloride. The term we use for a molecule that is moving outwards is efflux. So we will have potassium efflux. Efflux is the term we use when there is net movement outwards. The term we use for net movement inwards is influx. So for sodium and chloride, we constantly have influx. For potassium, we constantly have efflux. Remember, these are leaky channels, which means they're always open, which means potassium is constantly moving out of the cell. It also means that sodium is constantly moving into the cell. Chloride is also constantly moving into the cell. I'm expecting someone to then start asking themselves, if potassium is always moving out of the cell, at what point will it stop? Or will we deplete the intercellular fluid? So I've already asked that question for you. At the end of this lecture, you should have an answer to that question. Now, having agreed that those ions move in the directions that we have seen, we want to see the impact of those movements in terms of the charges on the cell membrane. Today you'll forgive my neighbor's dog. We want to see the impact of the efflux of potassium, the influx of sodium and chloride on the charges on the cell membrane. I hope you remember also that potassium is a cation, sodium is a cation, chloride is an anion. Now, we have said that generally potassium is more inside the cell than out of the cell. This blue region represents inside the cell. Then this is extracellular, this is extracellular. We have said that potassium will tend to move out of the cell. Being a positively charged ion, if potassium moves out of the cell, which means if we have potassium efflux, what does that mean in terms of the membrane charges? It means that potassium will be carrying positively charged ions out of the cell. I want you to grasp that. That as potassium moves outwards, it will carry with it positive charges out. If it carries positive charges out, it means that the inside of the cell will be more negative if you compare that with out of the cell. So what is the effect of potassium efflux? The effect of potassium efflux is that the inside of the cell becomes more negative, or we can say the outside of the cell becomes more positive. Anything that causes that kind of charge asymmetry is termed as being polarizing. Polarization is the making of the inside of the cell be more negative and the outside be more positive. So any ion that makes the inside of the cell be more negative is termed as causing polarization. Membrane polarization is when the inside becomes negative compared to outside. I hope you've understood that. That is membrane polarization. So potassium efflux causes membrane polarization. And I've defined polarization as when the inside becomes more negative, the outside becomes more positive. Right. Potassium efflux causes membrane polarization. Let's see what chloride does. And this one I want one of you to try. So the question is, chloride is a negatively charged ion. Chloride is more outside the cell. My question to you is, if chloride was to move through its leaky channels, will chloride movement cause polarization or depolarization? Anyone? And maybe the reason is more important than the answer. Anyone just try?
Maybe try.
Yes, Linda.
The chloride ions will cause depolarization too, because they also carry negative charges, making the inside of the cell to be more negative.
Okay, so you said chloride movement will cause polarization or depolarization? Which one?
Polarization.
Polarization. Thank you, Wamboi. So we are saying that chloride is negatively charged and it's more outside. That means if chloride leaks through its channels, it will be an influx. It will be carrying negative charges into the cell. And that means it will be making the inside be more negative and the outside be more positive. Therefore, chloride influx also causes membrane polarization. Now let's look at sodium. I want someone else to try. If sodium moves through its channels, what will sodium movement cause? Is it polarization or depolarization? Yes,
Depolarization.
it will cause
It will cause
depolarization.
depolarization.
All right, I agree with you. So, sodium, because sodium is positively charged and is more outside, if it moves, you are going to have sodium influx. That means the inside of the cell will be more positive. The outside of the cell will be more negative. So sodium causes the inside of the cell to be more positive and the outside of the cell to be more negative, because sodium is positively charged, is taking positive charges into the cell. That means that sodium does not polarize. Sodium influx does not polarize. As a matter of fact, sodium influx causes depolarization. So depolarization is when we make the inside be more positive, or if you make the outside be more negative. It's like you are canceling the polarization. So at that point, therefore, we can argue. We have things which cause polarization. We have things which cause depolarization. We can argue that then they'll cancel each other. But that's not true. The reason why that's not true is because in as much as we have factors that polarize and factors that depolarize, these ions do not diffuse across the cell membrane at the same rate. They do not diffuse at the same rate. As a matter of fact, when the membrane is at rest, potassium efflux is several times faster than sodium influx. Actually, about 50 times faster. That means that for every sodium that enters the cell, 50 potassium will have moved out of the cell. For every sodium that enters the cell, for every single sodium influx, there is 50 potassium efflux. Now, with that in mind, I want you to tell me, between polarization and depolarization, which one will override?
Polarization.
Polarization will override. I agree. Because in as much as sodium depolarizes, it is outweighed by the effect of potassium. So polarization overrides. Now, back to that important question I asked you. Your potassium is always moving out of the cell. There is a risk of depleting potassium inside the cell then. However, potassium is never depleted. Also, sodium is never depleted inside the cells. Sorry, outside the cell. It's never depleted. I want someone to tell me why. Okay, that could be a very difficult question, but maybe someone has already read. Why do you think potassium is never depleted inside the cells and sodium is never depleted out of the cells? Anyone? Just give a trial. It's something you can't.
Maybe I try.
Yes, yes.
Because you've said the efflux and efflux take place through the diffusion and in the leaky channels, and the diffusion is based on concentration. So when the concentration balance in that the outside and the inside have something like equilibrium, the diffusion will stop. There will be no more diffusion since there is a balance, and the concentration will be the same.
Thank you very much, Wamboy. You've really done well. So that's one of the reasons why you can never deplete. The fact that these are diffusions, so you'll definitely reach an equilibrium. Now, the other thing I want to tell you is that the equilibrium we are going to achieve here is not just based on the concentration of the specific ions. It is also based on the charges. When you have a lot of positive charges out, in as much as the concentration of potassium will still be higher here, and so it wants to move out, those positive charges will actually repel the positive potassium ions that are moving. So you'll actually achieve equilibrium of concentration, or rather, let me say it this way. The concept of equilibrium of concentration, as well as the concept of equilibrium of charges, will not really allow us to deplete potassium inside the cell. But then that makes us ask, so does it mean that it will reach a time that there's no movement at all? And maybe the answer is no. There's always movement. So why is it that there's always movement? That brings me to the second reason why we never deplete potassium and sodium in their respective compartments. Now, the reason is this. There is something we call the sodium-potassium pump. Sodium-potassium pump is something that pumps sodium and potassium against their concentration gradients. That means that it's a mechanism of active transport. It's a mechanism of active transport. How does it actively transport sodium and potassium? We know that sodium is more out of the cell. So if you're going to have active transport of sodium, it means that this pump will be moving sodium out of the cell. We also know that potassium is more inside the cell. So if you're going to have active transport of potassium, it means that this pump is going to cause potassium influx, not efflux, like what the leaky channels do, but influx. Now, this is what the pump does. In as much as it pumps those two ions, again, it does not pump them at the same rate. It will pump 3 sodium out for every 2 potassium in. Now, I want you people to look at that pump again with what I've told you about how it pumps. And tell me, what should be the effect on the charges, the net effect on the charges, if I take 2 potassium in for every 3 sodium out? Remember, sodium and potassium are both positively charged ions. Anyone?
Okay, I think the cell membrane will polarize.
It will cause polarization. I agree with you. So Nathaniel, why would the cell be polarized? What makes you say that sodium potassium pump polarizes the membrane?
Okay, this is because both the inside and the outside is positive.
Okay, that is not the definition of polarization though, Nathaniel.
Maybe I say, since you're pumping out 3 sodium ions, which means there are more positive ions being pumped out. So the outside of the cell will have more of the positive ions, causing polarization.
Very good. Thank you, Omboy. So that's the point. That this pump is taking 3 positives out for every 2 positives in. That means that the net movement is 1 positive out. It is causing positive charges net to be out, which means that there's net negative charges in. So the sodium potassium pump, because of the nature in which it is transporting the ions in question, it will also have a polarizing effect on the membrane. Good. I want you to remember something else. Okay, before we tell you what we remember, so we've said three things here so far: that potassium efflux causes polarization, chloride influx causes polarization, sodium potassium pump also causes polarization. Those are the three things we've said which cause polarization. In as much as sodium causes depolarization, the effect of sodium is outweighed by potassium. So we can ignore that for now. So because of these things, so far three things that cause polarization, we can say that the cell membrane is actually polarized at rest. One addition to add is that we mentioned that we have a lot of anions within the cell compared to out of the cell. And the anions we talked about are phosphates and proteins. Because we have a lot of negatively charged ions inside the cell, these ones also contribute to the inside of the cell being negative, which means they also contribute to the membrane polarization. Therefore, at rest, the cell membrane will be charged, with inside being negative and outside being positive. If that is measured, it will give us the resting membrane potential. So, if I was to summarize for you, 4 key factors that contribute to the resting membrane polarity, we will say one of the factors is this: that potassium efflux is there and is several times faster than sodium influx. So the effect of sodium influx is not felt really. The second factor is that we have sodium potassium pump that pumps 3 sodium out for every 2 potassium in, so the effect is that we have polarization. The third factor is chloride influx, negative charges entering. And the fourth factor is that we have more anions, negatively charged particles, within the cell than outside the cell. That means that at rest, the membrane is polarized. The membrane is charged at rest, and the charge is in such a way that the inside is negative, the outside is positive. Great. If you are to measure the resting membrane potential, we agreed it's measured in millivolts, but this measurement varies depending on the tissue. I will teach you 3 of them because this will form the basis of our next series of lectures in physiology. The resting membrane potential in neurons is negative 70 millivolts. The resting membrane potential in cardiac muscle is negative 85 millivolts. And the resting membrane potential in skeletal muscles is negative 90 millivolts. For the other cells, it varies, but not even important to know them because we'll not be talking about their physiology much. But these 3 will be talking of their physiology, and that resting membrane potential will be important to take into account. So remember these 3 measurements. Maybe the other thing I forgot to tell you is that all cells as long as they're alive, they will have a resting membrane potential. But the measurements I've given you are just for these three types of cells. Right. We are done with resting membrane potentials. Let me teach you about graded potentials before I give you some short break to think about something as I take the attendance. So, graded potentials, what are they? Graded potentials refer to transient disturbances on the normal resting membrane potential of a cell. You have a transient disturbance, which means a short-lived disturbance of the RMP. This disturbance only involves a small region of the cell membrane. The point here is it does not affect the whole cell membrane of that cell. It just disturbs a focus, a small region of that cell membrane. And that's the key thing. It is not diffuse. It is localized. It's focal. This type of disturbance can be either a depolarization disturbance, which means that if the resting membrane potential was negative 70, maybe it's now negative 60. You know, that's a depolarization because you are canceling the charges. It can also be a hyperpolarization, which means if it was at negative 70, maybe it's now negative 90. It depends on where we're starting from. So for example, if I give examples of neurons, neurons are at negative 70. If you have a stimulus that makes the cell membrane polarity to go towards minus 50, it means it is trying to depolarize because it's trying to make it go towards zero. That's depolarization. But if the resting membrane potential was at negative 70, then you have a stimulus that makes it even be more negative. We use the term hyperpolarization to mean it's making it more polarized. Hyperpolarization. So graded potentials can either be a depolarization effect or a hyperpolarization effect. Point is, it's a focal disturbance of the membrane. It does not affect the whole of the cell membrane. So the other parts of the cell membrane will just retain their normal resting membrane potential. If it's a neuron, it will just retain at 70. But at that point where there was a stimulus, you can have brief disturbance. So the amplitude of the disturbance will therefore depend, or rather, the amplitude of the amplitude of the disturbance will depend on the size of the stimulus. For it to move from minus 70 to wherever it's going, what will determine whether it reaches minus 80 or minus 90 is the size of the stimulus. So a weak stimulus will disturb it briefly, kidogo. But a stronger stimulus will have a higher amplitude of disturbance. That should make sense. If the stimulus is very weak, you'll just have a small disturbance. If the stimulus is very strong, you'll have a bigger disturbance. Great. I want to now talk about action potentials, but I want to give you a question first that you're going to do in your groups as I collect attendance. So the question I want to give you is to discuss the differences between graded potentials and resting membrane potentials. State the differences between graded potentials and resting membrane potentials. I'm dividing you into those groups right now. So there's something that will pop out in your screen. Just accept and be in one group. So I'm dividing you are 60 at the moment. I'm dividing you into 10 groups. You'll be 6 each. Something will pop in your screen. Just accept and be in your group. Right. I hope you've answered that question now. So in the next 20 to 30 minutes, we are going to talk about action potentials. And that will be the last part of the lecture. What are action potentials? Action potentials are rapidly propagated depolarization disturbances of the cell membrane. Get that right? It's rapid, which means it's happening very fast. They are propagated, which means they are moving along the cell membrane. It's not a constant thing. It's a wave that is moving. It's a depolarization, which means it is a time when the inside is positive, the outside is negative. That is a depolarization. Remember, polarization is when the inside is negative, the outside is positive. So, for those who may not have difficulty with the video, you can see in this neuron, there's a resting membrane potential where the inside is negative and the outside is positive. But there's a brief disturbance if you focus on at the level of the arrow, where you have positive charges inside and negative charges outside, but a brief one. That is a region of depolarization. But we also appreciate in this video that that depolarization is being propagated. In this one, it's moving from this region, the cell body of the neuron, along the axon to the end of the axon. It's a depolarization disturbance, and it is propagated rapidly from one end to another. Those are action potentials. What causes action potentials? Action potentials are caused by sodium influx. The other ion that can also cause action potential is calcium influx, but let's not talk about calcium for now. For now, let's just focus on sodium. Sodium influx is the one that causes depolarization, as we've seen when we were discussing the resting membrane potential. But that means that you must have a lot of sodium that overrides the effects of potassium efflux for it to cause a depolarization. And we are going to see that that actually happens. Now, that disturbance of depolarization usually spread along the cell membrane. So it is not like graded potential that is just on a particular focal point. This one spreads along the cell membrane. It's diffuse, so to speak. In as much as resting membrane potential and graded potentials are experienced by all living cells, action potentials are only experienced by muscles and neurons. Only muscles and neurons experience action potentials. Now, tissues which experience action potentials are termed excitable tissues. So action potentials are a reserve for excitable tissues, tissues which can generate action potentials are just muscles and neurons, nothing else. What is the importance of action potentials? Action potentials are important for transmission of signals. That means that we'll have signal transmission in neurons. We'll also have signal transmission in muscles. Those are the ones that can transmit signals along the cell membrane. These signals are electrical signals along the cell membrane. Now I want to see how action potentials are generated. Let's look at this. Consider this to be inside the cell, and this is outside the cell. The membrane at rest is positively charged out and negatively charged in. So this is the resting membrane potential. It's the resting state, normal for neurons. It's at -70. For muscles, it's at -85 for cardiac or -90 for skeletal muscle. Point is, this is the resting membrane potential. Now, we have sodium, and we know that sodium is more outside the cell than inside the cell. We also know that we have what we call voltage-gated sodium channels. Now, I'm bringing a new concept. When we talked about transport across the cell membrane, we mentioned that we have leaky channels, gated channels, and pumps. We've talked about leaky channels. Sodium leaky channel, potassium leaky channel, chloride leaky channels. We talked about them. Now I'm bringing the concept of gated channels. For gated channels, we said that we can have voltage gated, ligand gated, and mechanically gated channels. I'm bringing the concept of voltage gated sodium channels. We have voltage gated sodium channels, usually on the cell membrane, and that means that they're always closed, but they can be opened if there's a stimulus. Now, if we have a graded potential, that this causes disturbance on the resting membrane potential, that means that that graded potential is causing some voltage disturbance of the cell membrane. If that disturbance reaches threshold for a particular voltage gated channels, then it can actually open a voltage gated channel. Now in this case, if you have a graded potential that causes some voltage disturbance, it may cause the sodium voltage gated channels to open. Now, if you have opening of voltage gated channels, what will happen? You are going to have a massive sodium influx. If you open voltage gated sodium channels, you're going to have massive sodium influx. This massive sodium influx is unmatched. It cannot be cancelled by the regular leaky channels of potassium. Remember, for generation of RMP, we say that the leaky channels of potassium outmatch the leaky channels of sodium, and so polarization overrides. However, if you open voltage-gated sodium channels, depolarization will override. because these channels are very massive, very big, and you've opened them. So what will happen is that you're going to have sodium influx, massive, and that means that at that level you're going to have depolarization. What is important for you to note also is that as long as you've depolarized this region of the membrane, it means that the voltage gated channels here are also going to be opened, and that means you're going to have sodium influx somewhere there, and that will make the voltage gated channels here also to open, and you're going to have sodium influx again. Now I'm bringing the concept of propagation, that as long as you've opened voltage gate channels in a particular region, it will affect the neighboring membranes, and so it will affect the neighboring voltage gated sodium channels, and that means they're going to open, and so the depolarization is going to be propagated. Usually, during the depolarization, there's even a brief reversal of the membrane polarity. What I mean is that the inside become positive, the outside become negative. Remember, for normal state, the inside is negative, the outside is positive. At the peak of an action potential, the inside becomes positive, the outside becomes negative. You've reversed the polarity. It is important for you to note that the depolarization sequence spreads along the cell membrane to the end because of, as long as you have a disturbance in this segment, you're going to open, yes, this one has already been open, but you're also going to open voltage gated channels here because of the same effect, that there's a voltage disturbance that makes the voltage gated channels to open. And so when they open, they affect a membrane on a wider range, then that means that more voltage gated channels are opened again, and so the depolarization spreads throughout. That is why action potentials are propagated potentials. They spread throughout the membrane to the end. This is the concept of signal transmission. However, when the membrane is depolarized, it's a very unstable state for the cell membrane. Very unstable state for the cell membrane. See, the membrane is depolarized here. The depolarization is spreading less in that direction because this one is still the resting membrane potential. This is a very unstable state for the cell membrane. Very unstable state for the cell membrane. So what happens? When the membrane is depolarized, it will cause voltage gated potassium channels to also open. Now tell me what will happen if you open voltage gated potassium channels. It means that you are going to have massive potassium efflux. This massive potassium efflux is going to try to correct the membrane disturbance by making the inside be negative and the outside be positive. So basically, when you have this massive potassium efflux, you are trying to restore the resting state of the cell membrane by making the outside be positive and the inside be negative. So yes, potassium efflux will still come by opening voltage gated channels so that the resting state is restored. But either way, the sodium influx came faster, and so depolarization occurred, and it's even spreading along the cell membrane to the end. So the signal will be transmitted either way. Then now the cell membrane needs to go back to its resting state. This is the whole concept of action potentials and how they are propagated. Remember, the first one must be sodium influx that causes depolarization. Then later on, potassium efflux comes to restore the membrane to its normal state. Let's look at the phases of an action potential. And I want us to understand this graph. We can, if you are to draw a graph of the events that occur during an action potential, we will have such a graph. So the example given here is that of a neuron because you can see the resting state is at negative 70 millivolts. So let's take that to be the resting state, negative 70 millivolts. This region here represents the resting membrane potential. We have given the ionic explanation that is behind the resting membrane potential. We have said that the resting membrane potential is caused by 4 factors. Potassium efflux several times faster than sodium influx through their leaky channels. We've also talked about the effect of sodium-potassium pump. The third factor is chloride influx. And the fourth factor is that you have more negatively charged anions inside the cell. So that maintains the resting membrane potential at that level. If you have a graded potential that disturbs the membrane polarity, and that disturbance reaches a particular threshold. In this case, from this diagram, the threshold seems to be 15 millivolts. The difference between 70 and 55 is 15 millivolts. Usually the threshold can be anything between 10 to 20. So 15 is a good average. If the graded potential has an amplitude that enables the membrane to be disturbed by about 15 millivolts difference, then that disturbance can cause opening of voltage-gated sodium channels. If you have opening of voltage-gated sodium channels, you are going to have massive sodium influx. And that means that the membrane polarity, you are going to have a depolarization. So it's going to reach 0 and even make the inside be positive compared to the outside. So this phase of the action potential is what we call the depolarization phase. You can also call it upstroke. The stroke is going up, upstroke, or the depolarization phase. The depolarization phase of the action potential is due to massive sodium influx following opening of voltage-gated sodium channels. Now, when the voltage-gated sodium channels open, sodium ions will move in massively, and that will cause the membrane polarity to be depolarized. It will cause a depolarization. When the membrane is depolarized until the inside becomes positive, that has an effect on 2 things. One of those is that it will actually make the voltage-gated sodium channels to close. The moment the membrane polarity is reversed, voltage-gated sodium channels close. If voltage-gated sodium channels close, it means that we cannot continue having the upstroke. So we'll stop at some point. That peak of the action potential, where the pointer is right now, is what we call the overshoot. So what causes overshoot? Overshoot is caused by closure of voltage-gated sodium channels. Voltage-gated sodium channels close to cause that region there overshoot. At the same time, when you have the membrane in a depolarized state, it's a very uncomfortable state for the cell membrane. And so one of the effects it has is that it will make the voltage-gated potassium channels to open. If you have opening of voltage-gated potassium channels, you're going to have massive potassium efflux. Massive potassium efflux will make the cell membrane to now repolarize again. It will cause polarization. So we are using the term repolarization because it was polarized, then depolarized, so we are now repolarizing. And so this phase of the action potential is known as the repolarization phase. Repolarization phase is caused by massive potassium efflux following opening of voltage-gated potassium channels. Something interesting, when you open voltage-gated potassium channels, they remain open even beyond -70. The channels remain open. Usually, they will close at around -80. I want you to get that right, because I will ask you a question with regard to the pattern we see in neurons, the pattern we see in skeletal muscles, and the pattern we see in cardiac muscle, based on what I've told you. Voltage-gated potassium channels usually remain open up to -80. When it reaches -80, that's when they close. So in neurons, where the resting membrane potential is -70, when repolarization reaches -70, the voltage-gated potassium channels are still open. And because they're still open, it means that you are still going to have polarization. Now, when you're polarizing beyond the resting state, then we use the term hyperpolarization, or other is called the trough. So this phase of the action potential is called hyperpolarization, and is caused by the delayed opening of voltage-gated potassium channels. Delayed opening. They open in a delayed manner. They are still open. These are the phases of the action potential. Of course, if the membrane polarity reaches -80, the potassium channels, the voltage-gated potassium channels will close, and then that means that the factors that explain the resting membrane potential will come to be, and so the resting membrane potential will be restored. These are the phases of an action potential. So what I want you to take note is that in as much as we have hyperpolarization in neurons, we will not expect to have this in cardiac muscle, because the resting membrane potential for cardiac muscle is minus 85, somewhere here. So this potassium, voltage-gated potassium channels will close way before the resting membrane potential is established. And that also applies to skeletal muscles. Now I want us to finish by talking about properties of an action potential. One of the properties of an action potential is that an action potential can only be triggered by a depolarization. If you have a graded potential, we agree that graded potentials can either be a depolarization or a hyperpolarization. Hyperpolarizations cannot generate an action potential because, look at this. If this is the resting state, if you have a graded potential that is causing the membrane potential to go in this direction, hyperpolarization, that cannot cause sodium channels to open. It will not cause voltage-gated sodium channels to open. Voltage-gated sodium channels open only at around that level in this case. So if the disturbance is towards the direction, it will not make the sodium channels to open. And that is why we are saying it can only be triggered by a depolarization, not a hyperpolarization. The second property of action potentials is that action potentials have a threshold that will initiate them. Basically, what this means that there's a threshold voltage that can make voltage-gated sodium channels to open. That threshold voltage is between 10 to 20 millivolts. So if the resting potential was at 70, it means that if you disturb that resting potential to make it 62, it means you'll have disturbed by about 8. That cannot generate an action potential in as much as it is a depolarization. That disturbance must be between 10 to 20. So that means if the resting potential was at negative 70 and you make it be now negative 50 something, so the threshold here is within this range, that will cause an action potential. But if the threshold is kidogo like A and B, it doesn't reach the threshold. If the stimulus is low, it doesn't reach the threshold, this will not cause action potential. A and B cannot cause action potential because they are subthreshold. C and D are above threshold, so they can cause action potentials. The third property is that action potentials are all or none events. What that means is that you either have an action potential in totality or it doesn't occur at all. You cannot have a weak action potential. You cannot have a stronger action potential. It's an all or none event. It's either it's present or it's not present. So, like here, either there is no action potential or there is action potential. And if it's there, this will not be now bigger than this one because now this one was a bigger stimulus. No. It's either present or absent. Action potentials also have a constant amplitude. Now, this is the amplitude of an action potential. That amplitude is usually about 100 millivolts. Action potentials have a constant amplitude of about 100 millivolts, which means what? If the resting potential was at negative 70 here, we expect the overshoot to reach positive 30, so that the amplitude is 100. If the resting potential was minus 90 here, we expect the amplitude, the other side, to be at positive 10. so that we have an amplitude of 100. They have a constant amplitude, irrespective of the amount of the stimulus. So this is different from graded potentials. The other property is that during an action potential, there is reversal of membrane polarity. I told you that. This is at -70. There is depolarization with a reversal of membrane polarity. So here, the inside becomes positive and the outside becomes negative. You've reversed the membrane polarity transiently. The last one is that action potentials have a refractory period. Now let's look at this graph as I teach you refractory period. The refractory period is a period within which it is difficult to initiate another action potential. What that means is that when an action potential is occurring, and let's see from here, an action potential is occurring, it is difficult to initiate another action potential on top of this one. You cannot superimpose action potentials. So during this period where this action potential is taking place, the membrane is refractory to the generation of another action potential. Now, refractory periods are of two types. We have the absolute refractory period, where it is not possible completely to generate another action potential. So that applies for depolarization phase, overshoot, and repolarization phase. During those three phases, the cell membrane is completely refractory, which means you cannot generate another action potential. However, beyond this, during the undershoot, during the hyperpolarization phase, you can generate an action potential, but you have to use a stronger stimulus. And the concept is this. When the membrane is at hyperpolarization, it will take a lot of energy for this membrane to be disturbed up to this threshold. See, if it was here, -70, the energy that you'll use to make it reach -55 is less. But if you are at -80, the energy you need to make it to reach -55 is more. So this period here, the hyperpolarization phase, is a relative refractory period. You can generate an action potential, but you need more energy. More energy is required to generate an action potential during this phase. But during this phase, it is impossible to generate another action potential. These are the properties of action potentials. And so we are now done with the lecture. I don't know whether you have a question. So we'll now stop there. We are now done with cell biology. So our next series of lectures will be on basic tissues.